Well, welcome to the second video in this key area 2.2. In the last video we were having a look at ATP. And one of the things I didn't mention about it, which I'll just say just now, is that it's estimated that an active cell, something, for example, like a bacterium undergoing division, needs about something like 2 million molecules of ATP per second to satisfy its energy requirements. Now, it's actually found, though, that the quantity of ATP, say, present in a human body remains fairly constant, around about 50 grams, despite the fact that the body, as is indicated by just what a bacterial cell needs, despite the fact that the body needs a lot of ATP. Well, what is happening is that there's a very rapid turnover of ATP. As ATP molecules are being formed, as soon as that happens, they're undergoing a breakdown and releasing energy for cellular processes, and then at the same time ATP is being regenerated from ADP and inorganic phosphate, and so it's actually a fairly constant amount of ATP that's present in the cell or in our bodies, and this is because of the rapid turnover of ATP molecules. So moving on to have a look at the actual chemistry or the steps of respiration. Now, there are 3 reactions in cellular respiration that release the energy contained in food by oxidation, and I'll just talk about oxidation just in a wee moment, just to make clear for any of those of you who are known chemists. We're going to look at 3 stages. We're going to look at glycolysis, the citric acid, and the electron transport chain. Now we're going to look at glycolysis and the citric acid cycle in this video. So moving on, we need to remember that respiration occurs in every living cell. It's a chemical reaction or a metabolic pathway, and it involves the oxidation of food. And the energy that's released in that is used to regenerate ATP. Now I've put this slide up here just to talk a bit about oxidation. In oxidation, what is happening is that electrons are lost. And when we're thinking in terms of glucose, we can think of oxidation in terms of hydrogen being removed as well. So when we talk about respiration is an oxidative process. Hydrogen ions, electrons are being removed, and it's a catabolic, that is a breakdown reaction. Photosynthesis, on the other hand, is a synthesis reaction, and it's actually a reduction reaction. What's happening is hydrogens are being added. I suppose this is maybe a wee bit like a kind of biologist way of looking at it, but I think of it in terms of you've got glucose, a glucose molecule, and when that glucose molecule is being broken down, hydrogens are removed in that process. So that's an oxidative process. And when you're making glucose, you're making it from carbon dioxide and water, and so hydrogens are being added to form glucose. It's a wee bit of a basic way of thinking about it, and there's a lot more chemistry that's involved in that. So I will post a wee video on a chemist's view on all of this so you can get that viewpoint. But for the moment, if we just think of oxidation, or how if you've done chemistry, you think of oxidation in terms of loss of electrons. And that is what is happening in this situation here. So to sum up what's happening during the respiration pathway, well, your glucose is gradually broken down, and as that happens, hydrogen ions and electrons are released at various stages along the pathway. Now, what you need to know about is this enzyme, or there are a number of them, and they're called dehydrogenases, dehydrogenase enzymes, and they're involved, or they control the pathway of respiration. So let's look at the start of respiration. And the first part of respiration is glycolysis. Now, this occurs in the cytoplasm, if you remember that from your National 5. We'll talk about the mitochondria in a moment. Now, this diagram, the first part here shows your glucose and it being broken down to this molecule here, pyruvate. Now, in that process, you can see that 2 ATP are formed, Now, it's not showing you this in this diagram, but to actually start the process off, if you remember back to the video on ATP, then we talked about this being an energy investment phase. And what happens is that in actual fact, 2-ATP are actually, they are used to phosphorylate intermediates in glycolysis. There's 2 intermediates that are phosphorylated. In other words, the ATP gives a phosphate and an organic phosphate to them. And what then happens is that 4-ATP are actually produced. So because this series of reactions here produces 4-ATP, we say that there is a net gain of 2-ATP from glycolysis. So we're really talking about this being a sort of energy payoff stage here, where we invest 2 and out of that we get 2-ATP, if only the bank exchange mechanisms or bank interest mechanisms were as good as that. Now, at the same time, dehydrogenase enzymes remove hydrogen ions and high energy electrons, and they are passed to what is called a coenzyme. And in this case here, the coenzyme is called NAD. And these hydrogen ions and electrons are passed to that NAD, which is reduced to this form here, NADH. Sometimes you see it written instead of this part here, you see it NADH2. And that is what is known as your electronic sector. So by the end of this stage, we've got hydrogen and our high energy electrons, we've got our pyruvate, and we've got our 2-ATP molecules formed in glycolysis. Now, if there's oxygen present, then what happens is that pyruvate gets passed to the citric acid cycle. We'll talk in another video about what happens if there's no oxygen present, but at the moment we're going to talk about aerobic respiration and oxygen being present. So to do that, we need to have a wee look at mitochondria, because that's where the pyruvate is going to diffuse. Now, mitochondria have a double membrane. They've got this outer membrane here and also an inner membrane, that's this here. Now, on the inner membrane, that's where the electron transport chain is going to occur. When it comes to the citric acid cycle, that occurs in this area here, fluid-filled matrix. And it contains the enzymes that are involved in the citric acid reactions. So let's have a look at what goes on. Now, don't be over daunted by some of these pictures. You don't need to know all these formulas that are here. You just need to know the names. So our pyruvate diffuses from the cytoplasm into the mitochondria. And once it's inside the mitochondria, a carbon and 2 oxygen atoms are removed, forming carbon dioxide as waste product. And in that process, when the carbon is removed, a 2-carbon acetate molecule is produced. So we would be kind of round about here, acetate. Now that molecule is very small, so it can easily diffuse away. So there's a coenzyme A that is attached to it to form this here, which is called acetyl-CoA. And that's now ready for the citric acid cycle. And when this carbon dioxide is removed from the pyruvate to make the acetate, then you get extra high energy electrons that are produced. Now the NAD that is here, remember we talked about that before, it's an electron acceptor. It captures these electrons and it also attracts hydrogen ions to balance the charge, and that forms another molecule of NADH. So summing this up, really, what we can say is that pyruvate is broken down in this process to form acetyl-CoA, so 2-carbon molecule, and in that process NADH is formed and CO2 is lost. So we move on. Right, let's move on and have a look at the citric acid cycle. So whereabouts are we here? First of all, we're up here with the pyruvate. We just talked about this in the slide before, and we've formed our NAD, we've lost CO2 in the process, and we've formed our acetyl-CoA. Now that's a 2-carbon molecule. You don't actually need to know. I think the carbon numbers you used to in the old higher, but you don't need to know. But what happens is that this substance here, oxalo, that's an E there, oxaloacetate, I'm getting tongue-tied there, which is a 4-carbon molecule, it combines with the acetyl-CoA to form citrate. Citrate is a 6-carbon compound. Now what happens is that citrate then goes on through a series of enzyme catalase reactions back to oxaloacetate. Now because it goes from a 6-carbon to a 4-carbon, hence one of the reasons why I've written the carbons out, it's got to have lost some carbons along the way, and so it does. It loses them here in the form of carbon dioxide and here in the form of carbon dioxide to make the citrate back to oxaloacetate, and then the cycle will happen all over again, hence the reason why it's got a cycle. Now along the way as well, hydrogen ions are released along with high energy electrons, and they become bound to NAD. So here you see that here you see it happening here, and notice it over here as well. Now the hydrogen and the electrons can also bind to a different coenzyme. So NAD is one coenzyme, but you'll also notice that there's another here called FAD, and it is reduced by the addition of electrons to form FADH2, or the addition of hydrogens to form FADH2. So, and these coenzymes with their hydrogens, so NADH and FADH, will be used in the next stage for ATP production. You will notice a few boxes that I haven't filled in. They're actually, you don't need to know the names of these are just intermediate compounds. What you do need to know the names of are your pyruvate, your acetyl-CoA. You need to know about oxaloacetate. I'll just write these in black in case they're a wee bit hard to see. Acetate and your citrate. And you need to know that at the end of this process, really what you're ending up with are these NADs and these and the FADH2. Also as well in this, a molecule of ATP is produced. But what is important is the idea that you've got of these hydrogens and these electrons, because they are going to go on to the electron transport chain, and that is in the next video that we're going to have a wee look at. So I'll just fill in the intermediates here. And you should have in your notes, each of these stages, a note of what is happening as well. And then you can move on to have a look at the electron transport chain. I will also play some videos on iTunes U. They're maybe a wee bit more detailed than you need to have, but it's kind of useful sometimes just to have a wee bit of a background and understanding of what is going on.